CNA Exam Review 2024. Welcome to the quiz. Question 1. The sodium-potassium pump Na/KAT passe is fundamental to maintaining cellular resting membrane potential. What is its precise stoichiometry per ADP hydrolyzed, and what direct effect does its inhibition have on the intracellular ion concentration? Option A. Pumps 2 Na out and 3 K in. Inhibition causes intracellular hyperkalemia and increased negativity. Option B. Pumps 3 Na out and 2 K in. Inhibition causes intracellular accumulation of sodium and depolarization. Option C. Pumps 1 Ca2 out and 2 Na in. Inhibition causes intracellular calcium overload and hyperpolarization. Option D. Pumps 2 Cl out and 1 HCO3 in. Inhibition causes intracellular alkalosis and membrane stabilization. The answer is B. The sodium-potassium pump is an active transport protein embedded in the plasma membrane of most animal cells. It uses energy from the breakdown of ADP to pump ions against their concentration gradients. For every single ATP molecule hydrolyzed, the pump exports exactly 3 sodium ions Na from the cell and imports 2 potassium ions K into the cell. This 3:2 ratio is crucial because it creates both the ionic gradients high Na outside, high K inside, and contributes directly to the negative resting membrane potential by moving more positive charges out than in net export of 1 positive charge per cycle. If the pump is inhibited, e.g., by the poison Wabain or during severe energy depletion, it can no longer pump sodium out. Sodium ions will gradually leak into the cell down their concentration gradient and begin to accumulate inside. This buildup of positive sodium ions inside the cell makes the interior less negative relative to the outside, which is called depolarization. This disrupts the cell's ability to generate normal electrical signals. Option A reverses the stoichiometry. The pump does not import more K than it exports Na. Option C describes the function of a different exchanger, the sodium-calcium exchanger NCX, which is important in cardiac muscle and neurons, but is not the primary pump for establishing the resting potential. Option D describes an anion exchanger involved in pH regulation and has no role in establishing the Na/K gradients or resting membrane potential. Question 2. In protein synthesis, post-translational modification in the Golgi apparatus is crucial for protein function. For a lysosomal enzyme destined for the lysosome, what is the specific signal tag added in the cis-Golgi, and what is the receptor that recognizes it for correct vesicular trafficking? Option A. A KDL sequence retained by receptors in the smooth endoplasmic reticulum. Option B. A mannose 6-phosphate tag recognized by M6P receptors in the trans-Golgi network. Option C. A ubiquitin tag recognized by proteasomal complexes in the cytoplasm. Option D. A signal peptide recognized by the signal recognition particle SRP on the rough ER. The answer is B. Lysosomal enzymes are proteins that function inside lysosomes, which are acidic organelles that break down waste. To ensure these enzymes reach the lysosome and don't get secreted, they receive a specific molecular zip code in the Golgi apparatus. Enzymes in the cis-Golgi add a phosphate group to mannose sugar residues on the lysosomal enzyme, creating a structure called mannose 6-phosphate M6P. This M6P tag acts as the address. In the trans-Golgi network, there are specific receptor proteins called M6P receptors. These receptors bind to the M6P tag on the enzymes. Vesicles containing the receptor enzyme complex then bud off from the trans-Golgi network and deliver their cargo to late endosomes, which mature into lysosomes. The acidic environment of the endosome causes the enzyme to detach from the receptor, allowing the receptor to recycle back to the Golgi. Option A describes a KDL sequence, which is a signal that keeps resident proteins like chaperones in the endoplasmic reticulum ER, not for targeting to lysosomes. Option C describes ubiquitin, a tag used primarily to mark proteins for degradation by the proteasome, a different cellular disposal system. Option D describes the signal peptide, which is the initial tag that directs a ribosome making a protein to the rough ER. It is involved in the start of protein synthesis and translocation, not in Golgi-based sorting to lysosomes. Question 3. Oxidative phosphorylation is the final stage of aerobic respiration. Which complex in the electron transport chain is directly responsible for pumping protons across the inner mitochondrial membrane while simultaneously transferring electrons to molecular oxygen? And what toxic byproduct can form if this complex is inhibited? Option A, complex INAD dehydrogenase, leads to accumulation of succinate. Option B, complex 3-cytochrome BC1 complex, leads to superoxide radical formation. Option C, complex 4-cytochrome C oxidase, leads to the backup of electrons and increased reactive oxygen species ROS. Option D, ATP synthase complex V, leads to accumulation of inorganic phosphate. The answer is C. The electron transport chain ETC is a series of protein complexes in the inner mitochondrial membrane. Complex 4, also called cytochrome C oxidase, is the final complex. It has two critical jobs. It accepts electrons from cytochrome C and transfers them to molecular oxygen O2, reducing it to form water H2O. This electron transfer is tightly coupled to the pumping of protons H across the inner membrane, contributing to the proton gradient used to make ADP. If complex 4 is blocked, e.g., by cyanide or carbon monoxide, electrons cannot be passed to oxygen. This causes a traffic jam. Electrons back up along the chain. When electrons are stalled on earlier complexes like complex I and 3, they are more likely to leak and directly transfer single electrons to oxygen, forming superoxide radicals, a type of harmful reactive oxygen species ROS. Option A, complex I, does pump protons and is an entry point for electrons from NADH, but it does not interact directly with oxygen. Its inhibition would disrupt the chain, but the question specifies the complex that transfers electrons to oxygen. Option B, complex 3, also pumps protons and can be a significant site of superoxide production if inhibited, but again, it does not interact directly with oxygen. Option D, ADP synthase complex V, uses the proton gradient to make ADP. It does not pump protons or handle electrons from the ETC. Question 4. Apoptosis, or programmed cell death, is a regulated process. Which family of intracellular protease enzymes is the central executioner of apoptosis, and what is their mechanism of action upon activation by intrinsic or extrinsic pathways? Option A, caspases. They cleave specific cellular proteins, leading to controlled cell dismantling and phagocytosis. Option B, phospholipids. They degrade the nuclear membrane, causing lysis. Option C, kinases. They phosphorylate growth factor receptors, promoting survival. Option D, DNases. They randomly fragment DNA without energy requirement. The answer is A. Apoptosis is a highly controlled, non-inflammatory form of cell suicide. The central executioners are a family of enzymes called caspases. They exist as inactive precursors procaspases in healthy cells. When an apoptotic signal is received whether from within the cell intrinsic pathway, e.g., DNA damage, or from outside extrinsic pathway, e.g., a death ligand binding to a receptor-specific initiator caspases are activated. These then activate a cascade of effector caspases. The primary mechanism of action of these active caspases is proteolysis. They act like precise molecular scissors, cutting specific cellular proteins at certain amino acid sequences. They cleave proteins involved in cell structure like cytoskeletal components, DNA repair, cell cycle regulation, and they activate other destructive enzymes like DNases. This systematic, protein-cutting activity leads to the organized breakdown of the cell into small, membrane-bound fragments, apoptotic bodies that are neatly cleared away by neighboring phagocytic cells without causing inflammation. Option B, phospholipids, degrade phospholipids in cell membranes. This is associated with necrotic cell death, which is messy and inflammatory, not the orderly process of apoptosis. Option C, kinases add phosphate groups to proteins, typically activating pathways for cell growth, division, and survival, the opposite of promoting cell death. Option D, denases cut DNA. While DNA fragmentation is a hallmark of apoptosis, it is a downstream effect. Specific denases CAD/ICAD are activated by caspases. Caspases themselves are proteases, not nucleases, and DNA cutting is just one consequence of their activity. Question 5. The fluid mosaic model describes plasma membrane structure. Which component is primarily responsible for maintaining membrane fluidity at varying temperatures, and how does its proportion change in organisms adapting to colder environments? Option A, integral glycoproteins. Their number decreases to reduce permeability. Option B, peripheral proteins. They form a static cytoskeletal network. Option C, cholesterol. Its proportion increases to prevent membrane solidification. Option D, phosphatidylserine. It flips to the outer leaflet to signal apoptosis. The answer is C. The fluid mosaic model describes the cell membrane as a sea of phospholipids with proteins floating in it. Cholesterol is a key lipid molecule embedded within the phospholipid bilayer. Its primary role in membrane fluidity is as a bidirectional regulator. At high temperatures, cholesterol restricts the movement of phospholipid fatty acid tails, making the membrane less fluid and more stable. At low temperatures, it prevents the fatty acid tails from packing too tightly together, which would make the membrane rigid and brittle. Essentially, cholesterol acts as a fluidity buffer. In organisms like fish or bacteria adapting to colder environments, they increase the proportion of cholesterol or similar sterols in their membranes. This increase prevents the membrane from becoming too solid like butter in a fridge and maintains the necessary fluidity for proteins to function and for the membrane to remain selectively permeable. Option A, integral glycoproteins, are involved in transport, signaling, and cell recognition, but they do not primarily regulate bulk membrane fluidity. Option B, peripheral proteins, are attached to the membrane surface and are involved in signaling or structure, but do not affect fluidity. Option D, phosphatidylserine, is a specific phospholipid that normally faces the cytoplasm. Its flipping to the outer leaflet is a signal for apoptosis cell death, not a mechanism for temperature adaptation. Question 6. DNA replication is semi-conservative. On the lagging strand, what is the role of the enzyme DNA ligase, and what specific bond does it form between adjacent Okazaki fragments? Option A, it synthesizes RNA primers, forms phosphodiester bonds between ribonucleotides. Option B, it unwinds the DNA helix, forms hydrogen bonds between complementary bases. Option C, it joins DNA fragments, forms phosphodiester bonds between the 3-OH of one fragment and the 5-phosphate of the next. Option D, it proofreads the new strand, forms disulfide bonds between cysteine residues. The answer is C. During DNA replication, the two strands of the double helix are copied. The leading strand is synthesized continuously, but the lagging strand is synthesized in short pieces called Okazaki fragments. Each fragment starts with an RNA primer. After DNA polymerase extends the primer with DNA nucleotides, the RNA primer is removed, leaving a gap between the newly synthesized DNA fragment and the one before it. The enzyme DNA ligase has the specific job of sealing this gap. It does so by catalyzing the formation of a phosphodiester bond between the 3-hydroxyl-OH group at the end of one DNA fragment and the 5-phosphate group at the beginning of the next fragment. This creates a continuous sugar-phosphate backbone. Option A describes the function of primase, not ligase. Primase synthesizes the short RNA primers. Option B describes the function of helicase, which unwinds the DNA, and hydrogen bonds form spontaneously between complementary bases. No enzyme is needed to form them. Option D describes the proofreading function of some DNA polymerases, which have exonuclease activity, and incorrectly mentions disulfide bonds, which are covalent bonds between sulfur atoms in proteins, not in DNA. Great job. Don't forget to subscribe. Bye.